Good afternoon, everyone. Since I'm a relatively new face to the mesothelioma research community, I thought I'd introduce myself just a little bit more by sharing a little tidbit about how I arrived at the Pacific Meso Center last summer to help start up a new lab and a new research program. It turns out that Dr. Cameron and I are actually connected through the same network, going all the way back to the 1980s at the National Cancer Institute Surgery Branch. Now, back in the 80s, I was still climbing monkey bars and playing dodgeball in the schoolyards, and barely even knew how to read and write, and still learning how to do math at the time. But this is a network that comprises of some of today's most well-respected cancer immunology and immunotherapy researchers, and it's a network that mentored me through my PhD research when I was at USC in the millennial decade, just down the freeway from here. So last year, 2012, some 25 years after my schoolyard dodgeball days, I meet Dr. Cameron for the first time ever. And because of our common research backgrounds, it became clear rather quickly that both he and I share the same vision on how immunotherapy can be rationally combined with tumor cytoreductive therapies such as surgery and cryoablation to hopefully one day improve clinical outcomes for the average pleural mesothelioma patient. So I'm here now at the Pacific Meso Center, involved in something really unique and unprecedented to start up a freestanding mesothelioma research lab, and likewise, I think that the research that I'll be discussing today is also quite unique and provocative, and hopefully will open up new avenues for understanding mesothelioma biology and eventually contribute to the advancement of new treatment modalities to the clinic. So we all know that treating solid tumors such as pleural mesothelioma is frequently a daunting challenge. Some of the major characteristics of solid tumors that contribute to treatment resistance include avascular or ischemic regions in different areas of malignant tissue. This limits drug diffusion to these areas. Also, low oxygen concentration in these areas also limits their sensitivity to radiation. And ischemia in and of itself can induce molecular and cellular changes in different areas of malignant tissue, which further complicates our ability to thoroughly treat a tumor. Now, the solid tumor microenvironment is also known to be highly immunosuppressive as a result of a number of tumor-derived factors such as chemokines, immunosuppressive chemokines and cytokines, the expression of immunosuppressive ligands on tumor cells and the surrounding stromal components. Certain types of suppressive immune cells are also known to infiltrate into tumor masses, which further perpetuates the highly immunosuppressive environment within solid tumors. So how do researchers generally model solid tumors to learn more about treatment resistance mechanisms? So there are in vivo animal tumor models, mostly mice. These offer the complexity of living tissue and ethical issues notwithstanding. They're relatively expensive and laborious, very time-consuming to carry out animal experimentation studies. And in the end, there are still key biological differences between animal versus human tumors, which makes it difficult to translate preclinical research into effective clinical therapies. There are in vitro human tumor cell models, meaning tumor cells that are grown using tissue culture plates and incubators. These are derived from primary human tumors in the form of established immortalized cell lines, which grow perpetually in culture. Surgically resected primary tumors can also be diced into fragments and maintained in culture, but only for a finite period of time, which limits their overall utility for research. Now, most in vitro human tumor cell models are maintained in culture as suspensions or as flat two-dimensional monolayers, which really don't do that great of a job in simulating the complex three-dimensional structure and biology of primary human tumors. So what we've been using to model human solid tumors in vitro are three-dimensional multicellular tumor spheroids. This is also known as organotypic cultures, which offer an intermediary approach for modeling the three-dimensional complexities of primary human tumors, but outside of a living organism. These spheroids are assembled from established human tumor cell lines, and just like primary tumors, they actually grow in size over a period of about 1 to 2 weeks before they reach mechanical size limitations. But this time window offers us an opportunity to study human tumor biology in a manner that is more physiologically relevant than more conventional flat two-dimensional monolayers. So these spheroid cultures offer us a platform for bridging the tissue complexity gap between live animal tumors and primary human tumors. So this is, on the left, is a three-dimensional pleural mesothelioma spheroid constructed from a cell line called MSTO211H, which was originally derived from a patient's malignant pleural effusion specimen. And this cell line is normally maintained in culture as a flat two-dimensional monolayer with most of the cells nice and spread apart from the others. Now this isn't very tissue-like at all. Mesothelioma doesn't look like this in the body. It looks more like this, a round multicellular nodule that's more reminiscent of the primary pleural mesothelioma nodules that grow diffusely all over the chest wall, parts of the diaphragm, and in many cases the lining of the lungs. Another important characteristic of these spheroids is that they form necrotic centers due to nutrient and oxygen diffusion gradients, where the cells on the outer layers are well nourished and actively dividing, and the cells within the core, being deprived of nutrients and oxygen, become necrotic. And this is a hallmark characteristic of solid tumors in general, this sort of intratumoral cellular heterogeneity. So when we slice into these spheroids and examine them histologically, we can clearly see three distinct regions of tumor cells which mimic important characteristics of primary solid tumors. There's an area of actively dividing mitotic cells, there's a quiescent region, and then there's a necrotic area. Now, the overall biology of the tumor cells in these different compartments can vary significantly, so it's rational to hypothesize that the tumor cells within these different compartments may have different sensitivity to therapy. So what causes this cellular heterogeneity within a tumor? Well, a major factor is ischemia. The areas of a tumor that are situated further away from blood vessels are deprived of nutrients and oxygen. This alters their molecular and cellular characteristics. So we have to keep this in mind when researching new treatments, in that the overall biology of tumor cells in different areas of a tumor can vary significantly. But by using these three-dimensional multicellular tumor spheroids, we can now recapitulate this ischemia-induced cellular heterogeneity in vitro. As far as the molecular characteristics of three-dimensional spheroids, when we examine the overall gene expression signature of human mesothelioma cell lines that are grown as three-dimensional spheroids, we can see a significant number of genes that are upregulated compared to their corresponding two-dimensional monolayer cultures. Among these genes include those involved in cytokine receptor signaling, such as the interleukin-4 and TGF-beta receptors, a number of cytokines and chemokines associated with immune modulation and recruitment of suppressive immune cells into the tumor microenvironment, the VEGF genes associated with blood vessel growth, and the matrix metalloproteinases involved in tissue remodeling and the invasive capability of malignant tumor cells to invade into surrounding tissues. And all of these genes are known to have an important role in a variety of different human tumor types, including pleural mesothelioma. So what we're looking at here is a heat map, and from my angle, the colors don't show up as well, but what we're looking at is a heat map of differential gene expression, with the genes color-coded in the red representing high relative expression and the green representing low relative expression. And this type of analysis offers us a more global expression signature of a tissue specimen. And our working hypothesis moving forward is that when cell lines are grown as three-dimensional spheroids, this more closely simulates the signature of primary tumors arising within the body. So the next steps with this three-dimensional multicellular tumor spheroid model for mesothelioma is to validate their physiological relevance by comparing their overall molecular and cellular signature to clinical tumor specimens, and try to focus on the set of genes and proteins that are most significant in contributing to treatment resistance. So we'll continue with our gene expression analysis methods. As far as in situ protein expression analysis, we've been using fluorescence microscopy to learn more about the distribution and regulation of proteins in different areas of these three-dimensional spheroids. For instance, when we look at expression of TGF-beta 1, which is a multifunctional cytokine that has an important role in mesothelioma biology and also within localized immunosuppression, our preliminary experiments suggest that the expression of this cytokine isn't all throughout the spheroids. It's only expressed in certain compartments within their three-dimensional architecture. So the utility and versatility of fluorescence microscopy is that it allows us to assess wide assortment of factors that contribute to treatment resistance, whether it's hypoxia-associated factors or factors associated with radiotherapy and chemotherapy resistance. But what's of high priority to us right now is to understand the factors that contribute to immunotherapy resistance. And this type of basic understanding will be key towards developing immunotherapeutic regimens that have a higher likelihood of clinical success. So the solid tumor microenvironment is well known to harbor a wide variety of immunosuppressive factors ranging from infiltrating myeloid-derived suppressor cells and M2 macrophages, T regulatory cells, immunosuppressive cytokines, and nutrient-depleting enzymes, and the expression of immunosuppressive ligands by tumor cells and their surrounding stromal components. And all of these factors essentially shield the tumor from being eliminated by the immune system. Now, tumors did not invent these immunosuppressive processes. These are normal functions that already exist with an intact immune system. For instance, they protect healthy tissues from harmful autoimmune responses. They protect the developing fetus from being destroyed by the maternal immune system during pregnancy. It just happens that tumors microevolve within this immunological niche. Now, the field of immunology has learned a lot about the major immunosuppressive pathways that are active in more common cancers such as melanoma, kidney cancer, lung cancer, colon cancer. How much of that knowledge actually transfers over to pleural mesothelioma in particular? Well, that's not completely clear at this point, but there are some hints as to the major immune cells that influence tumor control in pleural mesothelioma patients. For instance, on the immunosuppressive side, it appears that M2 macrophages have a detrimental role, and increased infiltration of M2 macrophages into tumors correlates with poor patient survival, at least in non-epithelial mesotheliomas. So a logical place to start for research would be to learn more about the role of M2 macrophages and their monocyte precursors, how they are recruited into mesothelioma tumors, and when they get there, what specific signals cause them to differentiate into an immunosuppressive phenotype. On the other side of the coin, it appears that increased infiltration of effector CD8 T lymphocytes correlates with improved patient survival. So it does appear evident that the immune system does have a key role in dictating clinical outcomes in pleural mesothelioma patients. And if we can understand how we can alter the immunological environment within the tumor and blunt the effects of immunosuppressive cells such as M2 macrophages and tilt the balance in favor of anti-tumor CD8 T lymphocytes, then we may be able to rationally intervene therapeutically with immunotherapy to hopefully improve clinical outcomes for the average mesothelioma patient. So one approach to altering the immunological environment within the mesothelioma tumor area is to use immunostimulatory cytokines. Currently, there are two cytokines that have been approved by the FDA for treating cancer, interleukin-2 and interferon alpha. Neither of these are officially indicated for treating pleural mesothelioma due to lack of randomized clinical trials proving their statistical effectiveness for treating meso. There have been early phase clinical trials, small early phase clinical trials that have used these cytokines to treat mesothelioma, whether delivered systemically or by direct intrapleural injection. And there are some hints of clinical benefit, but again, not proven in large randomized clinical trials. There are dose-limiting toxicities associated with the use of cytokines, however, which limits our ability to deliver high doses to where it's needed most, which is within the tumor. For instance, with interleukin-2, it can cause a dangerous drop in blood pressure as a result of capillary leakage. With interferon alpha, there's some liver toxicity associated with its use, so it's challenging to deliver an effective dose of immunostimulatory cytokines to tumors while sparing normal tissues from toxicity. So the challenge that we face now is how to improve the therapeutic index of immunostimulatory cytokines by more specifically targeting them to the tumor area, extending the duration of their bioactivity while also minimizing side effects. So one provocative idea that we're looking into is to use a cellular carrier, such as mesenchymal stromal cells, to more specifically target immunostimulatory cytokines to the tumor area. Mesenchymal stromal cells are mesoderm precursors that are found in adult bone marrow, amniotic fluid, the placenta and umbilical cord blood of newborns, and in fat tissue. They are possibly immune privileged, although there's still some controversy regarding that. But if they are truly immune privileged, this would allow for tissue transplantation between unrelated individuals, which is what we call an allogeneic tissue transplantation. And there are ongoing clinical trials around the country utilizing allogeneic mesenchymal stromal cells to treat autoimmune diseases and also within the regenerative medicine space. So from a regulatory perspective, there is precedent for using allogeneic mesenchymal stromal cells to treat human disease. One of the normal functions of mesenchymal stromal cells is in tissue repair, so they're known to preferentially migrate to sites of injured tissue. And in many ways, a tumor is a form of chronic tissue injury. So these mesenchymal stromal cells are known to also have tumor trafficking properties, which makes them a possible carrier for targeted delivery of immunostimulatory cytokines to the tumor area, such as the surgical margins, where there are microscopic pockets of tumor cells left over, even after complete macroscopic resection of all visible primary tumors. So going back to our in vitro three-dimensional mesothelioma spheroid model, we now have a platform where we can evaluate and begin proof of concept studies to evaluate how modified allogeneic mesenchymal stromal cells can engraft and persist in three-dimensional tumor-like structures in vitro and their effect on surrounding tumor cells and associated stromal components. So specifically what we want to do is modify allogeneic mesenchymal stromal cells to produce immunostimulatory cytokines such as interferon alpha and graft them into these three-dimensional spheroids and then evaluate their effect on the overall immunosuppressive versus immune stimulation balance. So it's a lot of work ahead of us, but hopefully over the next year and by the time our next symposium rolls around, we'll have some promising data to report on this particular project. So I hope that wasn't too overly science-y and that everyone can take home at least a few concepts. These in vitro three-dimensional multicellular tumor spheroid cultures are a useful platform for modeling human mesothelioma tissue outside of a living organism. They allow us to study individual factors that contribute to treatment resistance. They also serve as a platform for early preclinical proof of concept studies of novel therapeutics. We still need to validate the model by comparing them with clinical tumor specimens and try to identify the most relevant disease-specific pathways that we're going to be able to study in this in vitro model. And lastly, the immune system does appear to have a vital role in controlling mesothelioma, and the design of optimal immunotherapeutic regimens should ideally focus on reducing immunosuppression within the tumor microenvironment while at the same time enhancing anti-tumor immune responses. So lastly, some acknowledgments to the wide breadth of scientific expertise that's associated with the Pacific Meso Center, both internally and externally as collaborators or advisors. My colleague in the lab, Irina Yanculescu, who handles, Irina Yanculescu, who handles all of the complicated genetics work that I touched upon a little bit earlier. A number of people at UCLA and the West Los Angeles VA. Mike Fishbein, who's our collaborating pathologist, and his technician Longshen Hong, Dongmai Ho over at the UCLA Punch-Worthington lab, Diana Gage, who's a radiation oncologist at the VA, Svetlana Kotova, a surgeon at the VA, Shervin Sharma, who's part of the tumor immunology program at UCLA, Marco Kostek and Warren Grunfest at the UCLA Bioengineering Department, who's, I guess Marco is next. He'll be talking about the development of a cryosphere to treat meso, and of course, scientific advisor to the Pacific Meso Center, Robert Cameron. Thank you.